Hello and welcome to GCSE Infection and Response with Mrs. Castanera. We're on the last lesson in this series, lesson 12. How do scientists develop drugs for use? So today's lesson 12 drug trials, and our keywords today are trial, blind trial, double-blind trial, placebo, optimum dose and efficacy. So to start with, what we're going to do is we're going to do the knowledge check. So if you could pause the video whilst you have a go at those questions, please, and then we'll go through them. Okay, let's go through those. Question 1. State where most drugs originate from. Most drugs originate from plants or plant materials. Question 2. Define the term drug. So a drug is a substance that has a physiological impact on the body. That means it makes changes within the chemistry of the body. Question 3. State what a painkiller does. So a painkiller reduces pain symptoms. Question 4. Identify what type of pathogen an antibiotic will work on. So antibiotics will only work on bacteria. If you've written virus or fungi or anything else, that would be incorrect. Only bacteria. Do make sure you're marking these in a pen of a different colour as usual, and that if you haven't written an answer, you write across first and then correct the answer, or you add in extra detail if you haven't quite got all of the information. Okay, question 5. Describe what can happen with the overuse of antibiotics. So we get increased amounts of antibiotic-resistant bacteria. Question 6. State in which year Dr. Fleming discovered penicillin. In fact, he rediscovered penicillin, as it says, 1928. Question 7. State how antibiotics work. Okay, so by antibiotics, they either kill or prevent the growth of bacteria. Question 8. State the 4 main microorganisms. So that is bacteria, virus, fungi and protist. So make sure you've marked all of those and done all the corrections, and then give yourself a score out of 8. Now we have had these questions before, so if you have got 5 or more, then well done. If you've got 4 or less, then really you need to review the previous lesson before continuing with this lesson. So we're going to move on. So today we're going to understand why new medicinal drugs have to be tested, and we're going to describe this clinical trial process for drug testing. So by the end of the lesson, we'll be able to do these things. So, development. Large pharmaceutical companies are producing new drugs on a regular basis. It's a long process before a drug will enter general circulation and be available to patients. Okay, so they have to do a lot of things before they are allowed by governments to be given to patients. So it's a 3-step process. Step 1, preclinical trials on cells and tissues. So step 1 doesn't go anywhere near any people, it just goes through cells and tissues that may belong to people and may belong to animals. Part 2 of the 3-step process is preclinical trial on live animals. Again, that's to see if it works, if it's safe, and if there's any side effects. Step 3 is clinical trials on human volunteers. So this procedure is essential for the safety of all patients. if we didn't do it this way, we could get some really dangerous side effects that could have really significant impact on people who take them. So now what I want you to do is I want you to watch these 2 videos, and they detail the steps involved in drug testing and that process of producing new drugs. And I want you to summarise these steps on the student notes page or pad of paper. So these are the links. They'll be in the document that you've already been sent, and you've been sent each week. So you can open that document in Word or whatever to access those links, or just type them out. So watch the 2 videos and then write some notes on the student notes page. You're going to write a little note to yourself to remind you to see your notes about these. Okay, so pause the video whilst you do that. Okay, so let's go through what preclinical laboratory tests look like. So this is the first stage, the lab tests, and that's stage 1, and it's a preclinical trial, and it's cells and tissue cultures in a laboratory. Okay, so we're looking for damage to individual cells, and we're looking to see what effect it has on the cells. So are the ingredients toxic to cells? Do they do the thing does the drug do what you expect it to on the cellular level in individual cells? So that's stage 1. Stage 2 is where we test the drug on living animals, such as small mammals. So usually rats or mice or other small mammals. And this is to test efficacy. Now, efficacy is whether it works or not, and whether it produces the required effect. So efficacy is a 2-part word. Does it work, and does it work how it's supposed to? As well as to test its toxicity. So how harmful is it? Is it toxic to animals, or is it not toxic? And so these 3 parts of stage 2 allow us to figure out how much of the drug we should use. So the idea is that we're looking to find out what's the best amount to use, and that helps us determine dose. Okay, so in the UK, we have a law that says that new drugs should be tested on 2 different animals. Now, there are some people who think that this is cruel, and other people think this is a way to ensure human safety, and so that no dangerous drugs can enter the system. So there are a lot of differing opinions on this, and animal testing is something you may well have heard of, but it's also something that at the moment is essential, and we don't really have an alternative to this at the moment. So until we can come up with real alternatives to this, to animal testing, that's the way it has to be done for now. Okay, so I've just written here, and I want you to write it as well. The animal testing is controversial, but currently we have no safe alternative. So let's look at clinical trials now. So clinical trials is the term used when we're testing a drug on humans, and the drugs will only get to this stage if it passes the safety checks of the preclinical trials, so stages 1 and 2. So clinical trials is human testing. Now, stage 1 of the clinical trials, so this is stage 3 of the whole process, but this section of this process. So stage 1 of section 3 of drug trial is that the drug can be tested on healthy individuals. Okay, so first we test it on healthy individuals. This is so that we can see about any side effects and not confuse them with symptoms. Okay, so if people are healthy and they don't have any existing illnesses, then any side effects that happen are clearly going to be side effects and not something that they would have got anyway. So the first dose, so that's the amount of drug given, is very low, and then slowly they increase it during the trial. And the idea here is to see, first of all, to check that at a low dose there are no nasty side effects, or any side effects in fact. And then as they increase the dose, they want to see what side effects occur and at how high a dose these side effects occur. And that allows them to figure out the optimum dose. So the optimum dose is the most effective with the least harmful side effects. And so optimum dose is what we're aiming for. So this optimum dose, you need to know what that is. That is what the whole point of clinical trials is about, to figure out the correct dosage, or the optimum dosage, which is most effective and least harmful. So they do need to monitor those side effects. So to ensure fair testing, which is really important, because if it wasn't a fair test, then the results wouldn't be reliable. So to ensure fair testing, our volunteers are split into two groups, and some are given a placebo. Okay, so a placebo is a substance that looks like the drug. In fact, it usually smells like the drug too, but it has no effect on anything. Okay, so it's really important that you understand what a placebo is. It's literally nothing. It's where patients who are given a placebo sometimes feel like it's making a difference, because actually being given medicine, whether or not it does anything, or being told something is medicine, in itself affects you and can make you feel better because it has a psychological effect. This is something you see typically in young children. So child scrapes its knee and then their mum or someone will say, oh, let me rub it better, let me kiss it better. And all these things, they don't really make any difference, but it makes a difference psychologically, which is in your mind. So it makes you feel better about it and feel like it's fixed. And that's why small children can often be calmed down from injuries or scrapes and stuff and cuts just by kissing it better, or rubbing it better, or whatever it is that would be done. So the whole point of this placebo in these two groups is so that the doctor can assess whether it's the drug making the difference or whether the person has improved because of the psychological effect of taking what they thought was a drug, called the placebo effect. So is it the placebo effect? They believed they were taking medicine so they got better, or is it because the drugs actually made a difference? Okay, we're going to write a bit more here about what the placebo is. Okay, so they felt better because they believed in the fake medicine. Okay, so the placebo effect is when someone feels better because they believed in the pretend medicine and nobody told them that it was pretend. So stage 2 of the clinical trials, so still in stage 3 overall, which is human clinical trials, but the second part of clinical trials is all about the patients who have the illness that this drug is designed to treat. So we get the patients who have suffering from an illness, they're going to test the drug on the people it's supposed to have an effect on. So these are called blind trials, and a blind trial is where the patient has no idea if they have the placebo or not. Okay, so the doctors in charge do know whether the patient has the placebo or not. In fact, it's more common for the trials to be double blind. So a double blind trial is where even the doctor doesn't know who's getting the drug and who's getting the placebo. And that's so that the company or organisation collecting the data can have completely unbiased data, because they don't want the doctors to subconsciously influence the results. And that can happen. So if you have a blind trial where the patient has no idea, but the doctor does, the doctor might ask questions of the patient. They say, so, you feeling better today? And they might say, oh, you're looking better today. How are you feeling? And so that's subconsciously suggesting to the person that they should be feeling better, because the doctor knows they should be feeling better, or is hoping that they do because of the drug. On the other side of that, if the doctor knows they're not having the drug, the doctor might say, oh, you look really uncomfortable, or you're in pain. How are you feeling? And they're just trying to be nice. They're not trying to influence the results, but that might encourage the person to say, oh yes, I am in a lot of pain, or oh yeah, I'm feeling horrible today. And so that's why usually these days we have double-blind trials where the doctor doesn't have a clue either. And that's because actually in the past doctors have subconsciously influenced those results in that way. So let's write a little note about what we mean by subconsciously influencing the results. Okay, so they are looking for improvements and convincing themselves there are some where there are not. There we go. So make sure you've got these written down. So doctors convince themselves and the patients that there are improvements in the patient's condition when actually there isn't. So despite no actual improvement. Okay, so we're going to move on to the next page. So before the results of trials are published, all cases, all drug trials, go through something called a peer review system. So before the results are published, it is peer reviewed. Okay, and we'll talk about what that is in a second. And that prevents false claims of a wonder drug. Okay, so peer review. So peer review is basically where your peers, which are people who are similar to you or at your level, review it. So this means that your results are checked by other scientists who have no interest or stake in it going well. So independent scientists check the process and they confirm or disprove the results of the trial. So peer review is when other independent scientists they check the trial results So it's when other independent scientists check the trial results and confirm or disprove them. And that's what peer reviewing is. And that prevents these false claims, as we said, of a wonder drug. So this stage of the testing system is really important, and it was implemented by many governments because of the effects that can happen with untested drugs. It's very important, and the effects that can happen with untested drug are really bad. And so we have an example of this, and it happened in this country, and it's really sad, okay? So this well-known example is called thalidomide. And so it's this drug that was initially prescribed as a sleeping pill, okay? It was given to pregnant women without being tested. And because it wasn't tested on pregnant women, it was only tested on regular women and everything was fine. They didn't know that it had an effect on the unborn baby. And actually, tens of thousands of babies were born with limb deformities. So they were born usually missing limbs or parts of limbs, so arms and legs. Some babies were born with none. Often they were born with really tiny limbs. So essentially, whilst they're in the womb, this thalidomide drug had prevented the arms and legs from developing properly. And so it would be a full-size baby or a full-size child with really tiny little barely formed arm and leg sort of deformities. And it was really sad because tens of thousands of women's babies, they were born with these serious deformities, and it had a lifelong effect on them, all because the drug wasn't tested properly on pregnant women. And actually at that time, what's more sad is that not only was the drug not tested, but doctors acted very independently. And so when each doctor would have a few women who'd have these awful limb deformities in their babies, they wouldn't connect the dots and realise that it's this thalidomide drug. And it took really a very long time, or much longer than it should have taken, for them to notice that these babies were being born with these deformities because of the thalidomide. It took several doctors noticing it and asking friends who were doctors, and then sort of it being discussed at a more large scale across counties and the country before doctors and the government realised that actually this was really serious, and there were a lot of women affected by it. It wasn't just these one-off occurrences. So it was really sad. It was really very sad. And there is a video on YouTube if you wanted to watch it. To be honest, you could literally type in thalidomide and you'll find it as long as you type it into YouTube. But I will put the link up for you on the next screen. Okay, so I'm going to write video link here. And that's optional, you don't have to watch it because it is distressing. And it tells you more about it. It doesn't tell you much about the science necessarily, but it does tell you more about it. And it's interesting, but quite sad. So we're going to move on to our checkpoint. So it's 3 questions here. One important test on drugs is for efficacy. Can you remember what efficacy means? You're going to need to know it. So describe what is meant by this term. So if you can define efficacy for 1 mark. Part B. Before clinical trials, drugs were tested in the laboratory. During laboratory trials, the drugs are not tested on people. Name 2 ways a drug could be tested during this stage of the trial. And that's worth 2 marks. So we're talking about drugs being tested in the laboratory, not on people. So how are they tested if they're not being tested on people? Part C. Clinical trials consist of several stages. So clinical trials, that's on people, several stages of testing. During the first phase of clinical trials, a drug is tested on healthy volunteers. Describe what happens during the next phase of testing. So stage/phase of testing. So after it's tested on healthy volunteers, what's the next stage of the testing? Okay, so that's 2 marks to describe that. So pause the video whilst you have a go at that, please. And then we'll go through the answers. Okay, let's go through that. So question A, the effectiveness of the drug or how well the drug works. That's 2 example definitions of efficacy. Part B, so you can have any 2 of the following. So it could be a tissue culture on cells, so testing it on tissues or they're testing it on cells. They might do computer modelling or they might do animal testing. So any 2 from these 3. And part C, they do it with a small number of patients or volunteers who have the disease being targeted. So first of all, you need to say that it's patients or volunteers, not lots of them, but a small number, so some patients, a few patients would be fine. And the second part is that they have the disease the drug is supposed to work with. So give yourself a mark for each of those. So it's out of 1 for the first one, out of 2 for the following, and then a total out of 5. If you've got 3 or more, you've done very well, well done. If you've got 2 or less, have another go at that, please. Or make sure you've written out, at the very least, all the answers, okay? And don't worry, you'll get better at that. So we're going to move on to the knowledge and application check. This is not the last stage in this lesson, unusually, because this is the last lesson of the series. So there are some activities to be done after this task. So you need to continue with the lesson, don't just stop it after this. Now, if you could complete the knowledge check, there are 10 questions and you will need to answer those. Pause the video whilst you do so. Okay, let's go through those questions. Question 1. State what a drug is tested on in preclinical trials. So that would be cells and tissues. Question 2. State who a drug is first tested on in clinical trials. So that would be healthy volunteers. And we say volunteers, and they do volunteer for it, but usually they are paid. Not lots, but a bit. Question 3. Explain your answer to the previous question. The reason is they're looking for side effects. And so the thing is that they need to know what the side effects are on healthy people so they don't get confused with symptoms of the disease, which people who they're trying to treat might have. Question 4. Define the term placebo. So a placebo is a drug that has no physiological impact. In fact, it's not really a drug. It's a pretend drug. Okay? So saying it's a fake or pretend drug will get you the mark. If you could add that it has no physiological effect, that would be very helpful because that's a good extension to that answer. So the green part is not necessary to get the mark, but very important. Question 5, define the term efficacy. So how good how good the drug is at doing what it's supposed to, so doing its job. You can also say how well the drug works. Okay, question 6. State what thalidomide was originally prescribed for. So it was originally prescribed as a sleeping tablet, I believe, but it also worked, it was given to pregnant women who were feeling very sick at the time. So it was a sleeping pill, but it was prescribed because it was so good at stopping morning sickness. Question 7. State 2 forms of transmission. So there's lots of options here. You can talk about direct and indirect. That's the 2 official forms. So direct contact is the one where you physically touch the bacteria on a surface or person, or the virus, whatever it is. Indirect is all the others, so that's water, air, food, and all the others, blood, animals, vectors, so there's a big list of them. Feel free to go back through and check that. Question 8, state what the job of a ribosome is. So this is quite a question from quite a while ago. The job of a ribosome is protein synthesis, so that means it makes proteins. Don't worry if you didn't get this, but this is something you definitely need to remember because they ask this question so frequently in the exams, so often. So, question 9. Give 2 things a drug trial aims to discover. So, the efficacy of the drug and the toxicity of the drug. So, how well it works and what side effects it produces. So, if you haven't used the word efficacy and toxicity, you can say how well it works and what side effects it produces. So that's absolutely fine to use different wording, but in future try and use the terms efficacy and toxicity. Now, question 10. Suggest why a drug designed to affect absorption of nutrients in the small intestine to the bloodstream will need to be tested on animals. Okay, so in this case, this question misleads us a little because it talks about what the drug does. So the drug's designed to affect absorption of nutrients in the small intestine to the bloodstream. Now, weirdly, this part is irrelevant to our question. Okay, this part you don't actually need to know, and examiners love to do this to you. So actually, the question really says, suggest where a drug will need to be tested on animals. Okay, so all drugs need to be tested on animals. If they're going to be for humans, okay, they have to be tested on animals beforehand. Why do we have to test drugs on animals? Well, that is to see if the desired effect occurs before we test it on humans. So that's to see if it works or not, or it produces the desired effect before it's tested on humans, and also whether or not it's dangerous. And although we talk about, how drugs have to be tested on animals, a lot of people don't realise how many drugs get scrapped at the earlier stages of testing, and there are a lot. So many drugs get rejected at the first phase where it's just been tested on cells. In fact, most drugs will be rejected multiple times at that stage before they ever get to stage 2. Stage 2 is the animal testing part, And even then, a lot of drugs, not all, but many drugs will be rejected several times as well at this stage because they have some nasty side effects. So make sure you've marked your work, and there are 10 questions. To be fair though, question 9 has got 2 marks, one for efficacy, one for toxicity. So give yourself a mark out of 11. If you got 7 or more, you've done exceptionally well because there were some tricky ones in there. If you've got 4 or less, you need to have another go through this lesson. Okay, so your extension activity is to research thalidomide and its effects. And in fact, let me double check. That is not the same video as earlier, so it's a different video to earlier. And you'll need to have a look at the keywords as well on page 39. So you'll need to know those. Okay, so make sure you understand what each of these things is. So what a blind trial is compared to a double-blind trial. Then, as the last tasks for this topic that will complete your knowledge on this is to complete the 30 questions synoptic assessment. Okay, and go through those. If you need an answer sheet or need your work checking, please discuss that with your teacher. Then you'll need to also have a look at and have a go at these questions, which are the application of knowledge questions on page 41. And those cover the whole topic. Okay, have a go at those. And then finally, you'll need to do the video review. Okay, so there is a video here where you can watch it on YouTube, and that goes through most of this topic, okay? In fact, all of this topic by the looks of it. So there is quite a few questions there, and it goes on to the next page, okay? And once you've done that, okay, watch the following video, and then you can mark and correct your answers. And if you're having any difficulty with any of the questions or work in any of that, we do have 2 revision set pages at the back of the book, page 44 and 45, which you can use to help you go through that content. Okay, so if you have any questions or any problems, please discuss that with your teacher, email them, or in class, that's fine. And that would be the end of this topic and the end of this lesson. Thank you very much.